This is a recording for Biology 1406, Unit 5, Chapter 11 from the OpenStax book, Meiosis and Sexual Reproduction. So sexual reproduction involves the recombination of cells called gametes in all organisms. The recombination of those cells and the combination of the genetic contents of those two cells, and the process of those two cells combining is called fertilization. The understood advantage of sexual reproduction for the organisms that do that is that novelties in the genetics of the two organisms can be recombined into additional new ways in order to increase the genetic diversity of the population, or specifically the genetic diversity of the offspring of those individuals, and therefore the genetic diversity of the population at large. So meiosis is the form of division, of cellular division, that produces gametes, and it is different from mitosis in that there are two rounds of division, and there is genetic recombination that results in cells being different from each other. The gametes are different from each other in meiotic division as compared to the daughter cells of mitosis are, barring accident or mutation, identical to each other. All right, so for sexual reproduction to occur, therefore for meiosis to occur, you have to have cells that have two sets of chromosomes. So that state is referred to as diploidy. Diploid as in having two copies of the chromosomes. So in this case right here represents the chromosome number, and 2 is going to be 2 times that number. So for instance, in human beings, the chromosome number N is 23. 2 times 23 is 46. And that's how many chromosomes that we have in each of our diploid cells, which is to say our somatic cells, the body cells. And this is quite true of eukaryotic organisms. The majority, certainly of the multicellular organisms, undergo sexual reproduction and therefore go through this process of meiosis to produce those gametes. And the reason we have to have this reduction division in this diploid state is that we take half of this diploid number of chromosomes and assign those to a gamete, and then we combine 2 of those haploid gametes together to recreate a diploid organism. So the life cycle is we alternate back and forth between diploid organisms, haploid gametes combined back together to create diploid cells that go on to develop into the adult organism. Oh, and also when we're talking about plants and fungi, but not animals, we also have a haploid cell referred to as a spore, which is distinct from a gamete in that a spore does not fertilize or fuse with another haploid cell to create a diploid cell. Instead, the spore germinates to create a haploid organism, and somewhere along the way that haploid organism produces specialized haploid cells, gametes, that fuse together to create a new diploid cell to recapitulate the life cycle. So spores exist in life cycles of organisms other than animals that undergo sexual reproduction. So that's fungi and plants and some protists. All right. So just comparing the two processes, mitosis and meiosis. Remember from our chapter on mitosis, when we were looking at mitosis, specifically mitosis refers to the division of a diploid nucleus into 2 haploid nuclei. And then, of course, that is followed immediately by cytokinesis, the division of the cell with 2 haploid nuclei in it to create 2 new cells with a nucleus inside each one. I said haploid in there for some reason. I didn't mean it. But mitosis can occur with either a haploid cell or a diploid cell. It doesn't matter. However many chromosomes are in the cell in mitosis, all those chromosomes are duplicated. The duplicated chromosomes are aligned for a division process, and then 1/2 of each of those duplications goes to either cell. And then we have our 2 daughter cells, which should be genetically identical to each other and effectively the same as the parent cell. In meiosis, this is only for producing these gametes, these reproductive cells or spores that lead to the production of sperm and gametes, through an intervening multicellular stage. Meiosis is also that division of the nuclear material, and necessarily it's followed by cytokinesis to produce the final divided cells. All right, so here we're looking at a flow chart of meiosis from a diploid cell. And remember, diploid just means 2 copies of each chromosome. So what happens in meiosis that's different from mitosis is after we go through interphase, you remember G1S, where we duplicate all of our DNA, and then G2, where we grow, or the gap, but it's I refer to it as growth 2, G2. The cell is ready for division, and then it goes into prophase. So the initial opening events in prophase are pretty similar. The nuclear envelope disintegrates. This envelope is gone. The chromosomes condense down from their unfurled chromatin back down into their condensed chromosome structures, the condensed X-shaped things that we're familiar with. Meanwhile, the centrioles have duplicated and start segregating from each other, producing a spindle apparatus between the 2 centriole pairs, or in plants, the centrosome regions. There's no actual centrioles in plants, just centrosomes. But the process is similar for any diploid cell undergoing meiosis. And so we're establishing that microtubule system for moving all the chromosomes to their final destinations. What's new is that our homologous chromosomes, so the duplicated 2 versions of chromosome number 1, the duplicated 2 versions of chromosome number 2, etc., etc., join together in what's called a tetrad, just means 4 chromosomes all associated with each other. And we start having chiasmata, which is where the arms of opposite chromosomes, so maternal and paternal, red and blue generally mean from the mother and from the father, but it doesn't matter. Just this is the chromosome from the parent of this cell. This is the chromosome from the other parent of this cell right here. And what we do is we exchange bits and pieces between those chromosomes to create recombinant chromosomes. It's not illustrated in this picture down here, but it is down here. You can see it here. Those recombinant chromosomes are the result of splicing back and forth pieces between the homologs, which means chromosomes with the same pattern of information in the same locations. We can have different versions. So for instance, the mother might have a gene for blonde hair right here, and the father has a gene for brown hair on the same chromosome that also, has a gene that codes for curly hair for the father and straight hair for the mother. And then we exchange pieces of these chromosomes. So now you create a new chromosome that might have wavy and blonde coded into it, and its opposite is going to have straight and brown coded into it, which would not be the same as either of the parents' genotypes. And that's the whole point of meiotic division and recombination, to create those new chromosomes carrying new combinations of features on them. So that's the big novelty. And then those homologous pairs are lined up on the division plate for meiosis 1, and the homologs are separated from each other after they've gone through this recombination process. So you get, one duplicated version of chromosome in this cell, the other duplicated version of chromosome in this cell, and note they've exchanged bits and pieces. That's genetic recombination called crossing over. And same thing for the other, in this case, chromosome pair, but for us there would be 23 of these pairs all in a line across here. So it's just complicated to make a drawing like that. And then, let's see if I can, okay, yeah, and that's the end of the first round of division. And then, and we'll have cytokinesis, so we'll produce 2 new cells. And then we go through another round of division, which is pretty much equivalent to meiosis. There's no practical difference between the two. So the second round of division in meiosis really is just mitosis. And that's where we take the duplicated chromosomes, line them up on a division plane, and split them again. Let's go ahead and take a look at a little more detail of meiosis 1, and then we'll go on to meiosis 2, the second round of division. So here's our duplicated chromosome. Remember, the centromere is a region that both attaches the duplicated chromosomes to each other and then also allows those chromosomes to attach to the microtubules of the spindle apparatus. The spindle apparatus right here. And then the homologous chromosome. So again, that means the two versions of chromosome number 1 that you have in you or in any organism. Red right here, the two red right here represent this one duplicated. This is a duplicated chromosome. Then blue is also a duplicated chromosome. And blue and red are homologs of each other in that they are the same pattern, same chromosomes carrying the same sort of information, but from two different sources, my father and my mother. The plant's father, the plant's mother, whatever it may be. And this is how we organize the chromosomes prior to the crossing over event. So then we're going to have crossing over between equivalent portions of homologous chromosomes. And, so A, B, C represent genes, just whatever particular genes right here. Lowercase represents other versions of the same genes. So those will be different versions of the same genes on the two different parents, so brown hair, blonde hair, curly hair, straight hair, whatever. These are just hypothetical. And here's that crossing over process. So once again, you can come up with a novel combination of alleles. The versions of genes are called alleles. So it's the same gene present in all 4 locations right here, coding for shape of hair. That's why these are homologous chromosomes. But we've got, in this case right here, one allele for the gene, one version of that gene that codes for one version of that feature. And here's a different version of that gene, a different allele, coding for a different version. The reason they're same on both of these sister chromatids right here is because this is a duplication of that. This right one over here is a duplication of this left one, or vice versa. It doesn't really matter. So that's why the duplications on a given chromosome are normally identical to each other. There is always the possibility of mutation, so that this might be different from that one there, but that's the exception rather than the rule. And so this is what we mean by recombinant chromosomes. All right. So it's the same steps and, roughly the same things going on in meiosis 1, but slightly different outcomes. So we create that spindle fiber system. The homologs are held together at these chiasmata. The nuclear membrane is completely broken down. All of those things happen right there. The chromosomes have condensed down into these things that look like this from the loose chromatin that they normally exist as. In metaphase, those homologous chromosome pairs are going to be aligned along the equator. All right, kinetochores attaching to microtubules leading off to the centrosomes and centrioles within them in the case of animal cells. And then here's an important part. The maternal and paternal chromatids align randomly on this metaphase plate. So that means the blue one on the right and the red one on the left, or the other way around. That's equally possible. There's a 50-50 possibility of which way each of these will be, this pair will be orientated, and the same thing for chromosome pair 2, not shown in this drawing, and pair 3 and pair 4 and pair 5. And there's no, the only process that's governed is one of them has to be on the left and one has to be on the right, or top, bottom, on either side of that metaphase plate. That's really the only thing that is required. And so that's not controlled in any other way. So each time meiosis occurs, you get a different pattern of which chromosomes are aligned left and right. And so you have many, many opportunities, in fact, 8 million novel ways in a human being in which you can combine one of each homolog pair in a given gamete. 2 to the 23rd is a nice big number that is 8 million and something. All right, and then at anaphase 1, we're going to go ahead and separate the tetrads. We don't break sister chromatids apart from each other, but we separate the homologs after crossing over has occurred. And then we go ahead and get them into a new, not a nucleus, because we don't reform the actual nucleus, but a new area, where all the DNA is gathered together, and then we split the cells in half to get 2 daughter cells. All right. A little bit about this process in plants versus animals. In animals, cells don't have a cell wall right here. We usually have an actin fiber ring that's what microfilaments are, is actin proteins. That kind of, this is the drawstring purse sort of analogy. These protein fibers are hooked onto transmembrane proteins, or at least integral proteins of the membrane on the inside of the cell right here. And so like if something inside um, is the actor, and that is the fibers pulling against each other with motor proteins and slowly pinching the cells in half, and eventually we get these two daughter cells. Plant cells that have rigid cell walls can't go through that process right there. So what they do is they bring vacuoles, transport vacuoles together that are full, that are by definition cell membrane, and then full of raw material to start putting together cell wall. And those vesicles fuse together to create an ever and ever larger plate of cell wall material. There's still communication between the two cells at the edges right here, and that plate is built and built and built until it joins, fuses with the existing cell membrane at both extremes right here, and the result is we've created two new cells where initially there was one cell. That structure is called a phragmoplast. Vesicles forming a cell plate. The entire thing is called a phragmoplast. One of my favorite words in biology, just because it's so odd. P-H-R-A-G, frag, M-O-P-L-A-S-T, plast. All right, let's go ahead and take a look at this animation right here. All right, so that just walked right through it there. Let's go ahead and do it step by step and see if we can make this happen. Let's see here. So we'll go to a diploid cell. So it's got 2 copies of each chromosome in it. In prophase 1, we see those chromosomes have condensed down, they've all duplicated, and now the homologs have joined together. And again, in this particular, for clarity, we just use a cell whose N number is 2. So 2N equals 4. There's 1, 2, 3, 4 duplicated chromosomes. There are 8 chromatids in here because every chromosome gets duplicated. You got to pay attention to that, those numbers there a little bit. But there are 4 distinct chromosomes here. 2 versions of chromosome 1, 2 versions of chromosome 2. The longest one's always 1, the next longest is 2. That's the rule there. And, blue and red represent maternal and paternal. Purple and yellow represent maternal and paternal, or back and forth, whatever you want to assign them to. We can see that the spindle apparatus is establishing, the centrioles have duplicated, and the nucleus itself is going away. In metaphase, we have the homologous pairs that are in tetrad form aligned on the metaphase plate. And notice here we've already got the crossing over events occurring. They happen a lot. The only regulation about crossing over is that it's supposed to be symmetrical. The same amount is swapped between equivalent chromosomes, and that it happens. And that it happens often. Those are the only rules, really, that we've been able to detect. Here we're dividing the duplicated, the homologous chromosomes from each other. The duplications are not being split yet. That is saved for meiosis 2. And then here in telophase, we see cytokinesis occurring at the same time. We're beginning to divide the cells in half. Then we reach prophase 2. The centrioles duplicate, create a new spindle apparatus again in each of the daughter cells. In metaphase 2, we align our duplicated chromosomes on the metaphase plate. So all of this looks just like mitosis. And that's the whole point. Meiosis 2 is just the mechanics of mitosis. After meiosis 1, which is where homologous chromosomes combine together into tetrads and swap equivalent pieces. And then the homologous chromosomes are separated from each other at the end of metaphase through anaphase and telophase. So this is just mitosis now. So here in anaphase, we divide the duplicated chromosomes in half, just like in mitosis. Here we get to telophase, and in the end, we've got 4 haploid gametes with, in this case, 2 chromosomes each. And the reason they look all fuzzy and puffy here is because the DNA is unraveling so that it is distributed again for reading. All right, so let's carry on right here. So here's meiosis 2, just in schematic form. So we're carrying on from this image right here. All right, so here's our separated duplicated chromosomes, one version of chromosome 1. The two sister chromatids are now not the same as each other. That's kind of important. Same thing for chromosome 2. And then it's the exact inverse. Whatever we've got here, it's going to be the reverse over here in this other cell right here. It's one, version of chromosome number 1 duplicated, but with the crossing over events making the two duplications dissimilar from each other now. Anyway, they line up on the metaphase plate. As I said, all of this looks just like mitosis now. The duplicated chromosomes are split in anaphase and segregated to their new nuclei in telophase, and then we have cytokinesis, and the nuclei will reform. And we get 4 haploid gametes from one diploid parent cell. So that's an important number. How many cells are produced during meiosis? 4 daughter cells from a single parent cell. So there are some details, but we don't really need to worry about them. The key things is the duplicated chromosomes are going to attach to that microtubule system. They're attached right here, and then they're going to be split apart. This is meiosis 2 right here. Metaphase 2 over here, anaphase 2 over here, and this is metaphase and anaphase 1. All right. All right, I think I've said all of this, so I won't redo this again right here. But you go back and listen or read this material here. All right, so here's a comparison side by side of the two processes. Dividing the homologous pairs, not the duplicated chromosomes, assigning those homologous pairs randomly, left or right, who ends up in this cell, who ends up in that cell, 2 to the 23rd different unique ways in which that can occur. Let me show you the math really quickly. So we've got 2 ways in which each pair of homologous chromosomes can be orientated, paternal to one cell or maternal to that same cell. So that's where the 2 comes from. And then we have 23 pairs of chromosomes in our cells. Other organisms have different. So we're going to take this and run it exponentially. So x to the y, and y is going to be 23. And that equals 8,388,608 unique combinations of chromosomes just from you due to random assortment that you produce in your gametes. All right, so the odds of making the same gamete twice just because of random assortment of the pairs of chromosomes is 8.4 million to 1. That's pretty long odds. And then if you add the crossing over, mixing of the bits and pieces of chromosomes together, the odds of making the same gamete twice for you, for an individual, is such huge numbers. It's going to be in the trillions that it's not even really worth calculating anymore. The odds of making the same gamete twice are astronomically against. If you include both crossing over, the exchange of genetic material between homologous chromosomes, and then the random assortment of which chromosome pairs on the left or right at the first split at meiosis 1, at anaphase 1 specifically, how they end up lining up on the metaphase plate at metaphase 1. After this, again, meiosis 2 is really just mitosis of a haploid cell, and we produce 2 cells that are nominally identical to their parents right here. But remember, the chromosomes, the duplicated chromosomes, are no longer identical here because of the crossing over event that happened back here. None of that, of course, occurs in meiosis. In mitosis. All right. So sexual reproduction is typical of plants, insects, fungi, quite a large number of protists, basically most eukaryotic organisms, not all. And there are plants that are not sexually reproducing, there are animals that are not sexually reproducing, and quite a lot of protists that aren't. And then quite a few organisms that kind of switch back and forth between sexual and asexual reproduction. Mitosis-based reproduction and meiosis-based reproduction, which is sexual reproduction. Just depends. There's lots of variation out there, but most organisms do indeed, most eukaryotic organisms do indeed undergo sexual reproduction. And the reasons are for that genetic recombination, giving them a evolutionary edge by creating more variation in the population for natural selection to take advantage of. At this point, we want you to learn 3 different life cycles, which is the sequence of going between haploid and diploid that is characteristic of sexual life cycles. So all sexual life cycles involve a diploid and a haploid state, cells that are in one state or the other. And we're going to look at animals, we're going to look at fungi, and we're going to look at plants right here. These are male pine cones, by the way, right here. Not female, but male pine cones. This is a toadstool, which is a diploid structure created by haploid fungi, and this of course is a dragonfly. It's a diploid organism just like you and I. This plant right here is a diploid organism, but within these male cones are little haploid organisms that produce gametes. All right. So we've got these 3 different life cycles. There's one that's diploid dominant, that's animals. There's one that's haploid dominant, that's most fungi. And then there's one that is, both of these exist as nominally dominant forms, and that's alternation of generations, which is typical of plants. So let's look at the diploid dominant life cycle as well. This is most animals right here. All right, and let me go to the picture and we'll go ahead and, yeah, there we go. Oh, no. So here's an image of a diploid dominant life cycle. And what diploid dominant means is that it's a life cycle, it's a sexual life cycle. So of course meiosis occurs, but mitosis only occurs in the diploid state. So that is the fertilized egg dividing via many, many, many rounds of mitotic division to create an adult animal. And this is true for all animals with some variation. And that's where mitosis occurs. So mitosis occurs, but only in the diploid state. That's what's meant by a diploid dominant life cycle. So in the gonads, called an ovary for producing eggs and a testis for producing sperm, in the gonads we're going to have diploid cells from this diploid organism, you and me. But we're going to have diploid cells within those organs that divide via meiosis to create haploid cells, and those are going to be the unfertilized eggs and of course the sperm cells right here. And then the only purpose of those haploid cells is to find their counterpart, or specifically for the sperm cells to locate the eggs, either through external fertilization in the water for aquatic organisms, a lot of them, or through some means of directed movement of sperm to eggs in terrestrial settings. Oftentimes internal fertilization in animals. Anyway, and then the cells fuse, the sperm introduces this genetic material into the egg. Those 2 sets of haploid genetics are combined together to create a new diploid cell, the fertilized egg, called a zygote, and then we're off running again. And that's the alternation, that's the generation. That's the life cycle, of an animal in general. Now the haploid dominant life cycle is the exact opposite, and this is typical of fungi and a lot of algae. And so the thing that you're looking at when you look at a fungus is typically a haploid organism. It produces gametes via mitosis. Those gametes fuse to create a zygote, and then that zygote, that duplicated cell, does not go through mitosis because it's a haploid dominated life cycle. Instead, that zygote, that diploid cell, immediately divides via meiosis to produce haploid cells called spores, and those spores germinate and grow via mitosis to make the new organism, the new fungus. All right, so here we're looking at an example. We haven't studied fungi really, but in lab you've seen single-celled fungi, primarily yeast. Here we're looking at multicellular fungi, but yeast do the same thing. And so here's a hypha, the filament that is the characteristic of fungi. You know, if you rip open rotten logs, you see the hyphae, little white filaments all inside there. Some of your food goes off. It might be covered in a brown, or sorry, in a blue-green sort of fur. If you look under a microscope, you'll see that that fur is really these hyphae looking like this. And that's the way fungi look. Anyway, those cells are haploid. They germinated from a spore. The spores let go by a mushroom, for instance, or a toadstool, or a bracket fungus, or something like that, or indeed the dust on that rotten orange with the, you know, the green fungus on it. And those spores divide via mitosis in the haploid stage, right here, to make a multicellular organism, the actual fungus, right? And a lot of fungi can reproduce asexually and do that quite often. But quite a few of them also at some point will go into a sexual reproduction process where they produce specialized haploid cells, all right, that are going to be the equivalent of gametes. And those gametes from two different cells, you know, from two different fungi, you know, one and another one over here, they find each other. The two fungi grow to each other. They have cells that attach to each other, and then they fuse these nuclei to create diploid nuclei inside this special structure right here. And that's the thing that's inside a mushroom in the gills of a mushroom. And then there we will have meiosis. The only thing these diploid cells do is divide via meiosis to produce spores. And so we've drawn 4 spores here to be the products of meiosis of just one of these diploid cells in here. There's a whole pile right here, so all of these, each of these can make 4 spores in this sort of image right here. And that's the haploid dominant life cycle. So it's really the exact inverse of the diploid dominant life cycle. And again, that's typical of fungi. Now, an alternation of generations, which the rest of the algae that are sexually reproducing, and certainly all plants undergo, all sexually reproducing plants, is alternation of generations. And the big difference here is that mitosis occurs both in the haploid and in the diploid state. Otherwise, everything's fairly, very much the same. So let's look at that life cycle here. We've got a lot of pictures which add variety to it, but let's just pay attention to, the ploidy and where we have mitosis. So we'll start here with spores, because all plants still produce spores one way or another. In 1407, you'll learn about differences amongst groups of plants. And, in other advanced courses, you would learn more. But anyway, as a generalization, all plants produce haploid spores. They're the product of meiosis, just like in fungi. So this is a haploid cell. And just like fungi, the haploid spore can germinate. So that is mitosis. Mitosis is occurring here to produce a multicellular plant that is haploid. And then somewhere on that haploid plant, we're going to have some specialized cells and structures that produce a haploid egg, and somewhere else, the male equivalent that produces haploid sperm cells right here, flagellated sperm cells right here. And just like in animals, the flagellated sperm must find the egg one way or another, and that's the fertilization event. And then we have that fertilized zygote. Now, if this was fungi, this would immediately go to meiosis and produce spores, but it's not. It's a plant. It divides via mitosis to produce a diploid plant called a sporophyte. That is phyta is plant and spore is spore. So this is a plant that produces spores. This is a gametophyte. That is a plant that produces gametes. And eventually that sporophyte will have some structures that are going to go through meiosis to produce haploid cells. In a way, you could think of these as being kind of like the ovaries or the testes on an animal, in that they are diploid cells that produce via meiosis some haploid cells. But instead of these being gametes, they are spores. And we produce this entire multicellular haploid thing from a spore, and then it, in turn, produces haploid gametes that fuse together to recapitulate the life cycle. So that's alternation of generations. And the big difference here is that we've got mitosis in the haploid stage, and we have mitosis in the diploid stage. All right, let's take a look at the study guide and add in anything that's missing right there. So I think we've defined asexual reproduction. That is reproduction that relies solely on mitosis to create new cells for the new organism, and that's exactly what happens. We call it something different, but it's still effectively mitosis in bacteria, in prokaryotic cells, and that's the only way they can reproduce. And then lots and lots of eukaryotic organisms can also reproduce by mitosis, lots of protists, one cell giving rise to two new cells that are nominally identical to each other. And in multicellular organisms, think about a plant, that creates a runner coming off of it that roots itself in the ground and then establishes itself as a fresh new plant, and that is via mitosis. Sea anemones, if you're not aware of it, can split in half to create two new sea anemones. And that's asexual reproduction, and mitosis is what's driving all of that, the dividing of cells that are the result of mitotic divisions. And of course, we've just talked a whole bunch about what sexual reproduction is. So that is what meiotic division is all about. A clone is another name for the products of mitosis. So, the clones are theoretically genetically identical to each other. And I always say theoretically because there's lots of little other things that can make this change, including random mutation during DNA duplication. Budding is a means of separating of reproduction for things like sea anemones and a lot of plants. Simple animals and lots of plants can reproduce asexually by budding. Interestingly, in the world of animals, there's quite a few organisms that are diploid. There's a bunch of fish, a bunch of reptiles, lots of insects, and currently a bunch of arthropods. There's a lot of things. No mammals that I'm aware of normally, but that entire species are parthenogenic, and what that means is they're diploid. All the members of the population are female, and the females produce diploid eggs. So it's always diploid. And they produce diploid eggs. And those eggs are fertile and divide via mitosis to create a new individual that's effectively a clone. Creating clones. And so there are parthenogenic species that are all female, and all of the females are effectively clones of each other, except of course mutations that still occur during duplication of DNA for mitosis. A lot of animals, especially ones that have a hard time finding mates and have limited sensory abilities, you'll find that the individuals actually are hermaphroditic, and that means they contain gonads for both genders at the same time. So they have an ovary and they have a testis, and they have the means of producing and delivering those cells. Now, hermaphrodites normally can't fertilize themselves. That would not be useful. Instead, both individuals can fertilize each other when they mate. So all snails, to my knowledge, are hermaphrodites, and lots of other animals. Earthworms are hermaphrodites. And so that's when you see them mating, they are taking on the role both of male and female, and both can get fertilized from the other and then lay eggs in turn. So, the advantage of a hermaphrodite is both individuals, any individual of the species that's met is automatically a candidate mate for mating. We've talked about haploid, diploid, mitosis, and meiosis, so I think that's pretty good. Reduction division is the proper term for the divisions that occur in meiosis. So meiosis and reduction division are kind of synonyms for each other. This means reducing the chromosome count when you divide, and that's what happens in meiosis 1. All right, we've talked about sister chromatids. That equals duplicated chromosome, and homologous chromosomes, two versions from two different parents. All right. A karyotype, let's not worry about that. I'm just going to gray that right out. not going to really worry about karyotype. Chiasmata and crossing over have to do with the crossing over process. Chiasmata is the overlaying of two different homologous chromosome pieces so that the crossing over event can occur. Crossing over is the actual snipping and recombining of the bits and pieces. All right, then of course go through the steps of mitosis, go through the steps of meiosis. All right, some concepts that we want to address right here. Costs and benefits of asexual reproduction and sexual reproduction. All right, so asexual equals cheap, fast, but no added variation for natural selection to act upon. All right. So the benefits are that it's cheap and fast, but the bad things are there's no added variation for natural selection to act upon. And so if we look at sexual reproduction, we're going to get the exact opposite. Whoops. It's going to be slow, expensive, comparatively. You have to find a mate, you have to have reproductive organs. You're slowed down because you have to develop those reproductive organs, you have to find a mate and all of that sort of stuff. But there is much added genetic variation in the population for natural selection to act upon. The costs and benefits of each. And so if this is a winning combo over here, things will reproduce asexually, and there's a whole bunch of things that do that. If this is a winning combination over here, things will reproduce sexually, and there's an even bigger number of things in the eukaryotic world that do this. And interestingly, there's quite a few living organisms that swap back and forth. Many plants reproduce asexually and can reproduce sexually, and they do both. So they can reproduce quickly, cheap and fast often, and then periodically, episodically indulge in sexual reproduction to get this added genetic variation. And so that's kind of really why some organisms can do without sexual reproduction, and why some organisms must employ sexual reproduction. The reason sexual reproduction is enjoyable, like in animals, like in us, is to drive us to obviously do this because it's beneficial for the species. Anyway, why can some organisms? So, a lot of small organisms with very short lives and very, very short generation times. Think of bacteria in minutes. Think of a lot of eukaryotic protists where it just may be a few hours to make a new generation, like yeast and stuff like that. Those organisms reproduce so quickly. They have so many generations in such a short period of time, and produce such large numbers of individuals that they produce enough genetic variation just through mutation to adapt to a changing world. That's pretty much it. All right. Why must some organisms employ sexual reproduction? Long-lived, slow generation time, small numbers of individuals require more genetic variation in order to keep up with changing world. All right. So let me highlight these here, right? All right. So, for instance, let me give you an example. E. coli bacteria, or not. Let's not give E. coli a bad name, but um, um, Bacillus pneumoniae. The organism that produces pneumonia. It's a bacterium, all right, or which one of them is a bacterium. Because it's a bacteria, it gets enough genetic variation to rapidly adapt, to both changes in the world and particularly changes in the host organisms that they infect, so that they can keep ahead of the changing immune systems and various other adaptations of those organisms, so they can keep infecting things. Well, a eukaryotic, multicellular, slowly reproducing thing like a human being, we live about 100 years as opposed to, maybe a few hours for a bacterial cell, have slow generation times, let's say, typically 22 years or so before we produce an offspring, maybe more these days, 25, I think average for the US now. Whereas these things can reproduce an offspring cell in a matter of minutes oftentimes. And these, can quickly go from 1 to millions, while in the same amount of time that we got millions here, we haven't even reproduced once for us. So we need the extra genetic variation that sexual reproduction offers to allow us to adapt to these disease organisms and at least, produce individuals that may be relatively immune to these disease-causing organisms. So that's the kind of arms race that we're in. And then also we have to adapt to the changing environment. Humans, not so much, because we're controlling our environment, but certainly all other living organisms, changes in temperatures, water availability, and all that sort of stuff. The bigger and slower you are, and longer lived, the more important sex becomes, sexual reproduction. All right, make sure that you can go through the steps and bits and pieces of mitosis and meiosis. Make sure that you can diagram for the test or answer questions about a diagram on sexual life cycles. What are the 3 sources of genetic variation that are unique to sexual reproduction? Those are crossing over, random assortment, and then random fertilization. The fact that we produce millions and millions of sperm cells in an attempt to fertilize one egg is an example of random fertilization. So which one of those, each quite unique spermatozoa, fertilizes that egg is another source of genetic variation. And the idea here is that the same combination is extraordinarily unlikely to occur twice. What's the ultimate source of genetic variation common to asexual and sexual reproduction? Mutation. All right, so that's our study guide. Here's your homework. It's fairly obvious. This will be handed in before you take the final. All right. And I've got a puzzle. When we look at these 3 organisms whose life cycles you have to draw, there is a historical reference, and I give you a clue. It's the middle of the 20th century. There's a kind of a pun, a historical, thing that these 3 genus species names could lead you to if you're curious. If you figure it out, I'll drop a homework grade. If you submit the answer to this puzzle right here in your homework. Tell me that you've done it so I remember to look for it. All right, that's going to be it for meiosis and sexual reproduction.